Welcome to Chapter 17, From Gene to Protein, Part 1. So in general, it's important to know the flow of genetic information. The process of gene expression, which is how you get from the original DNA to the actual proteins being made, has two phases. First transcription and then translation. Now typically, one gene makes one protein. Technically, one gene actually makes one polypeptide, and some proteins are made up of multiple polypeptides. Some polypeptides are even chopped down into multiple proteins, but scientists often use imprecise language and say protein when they really mean polypeptide. So when we say So, the first phase, transcription, is the synthesis of RNA using DNA as a template. So here's a diagram of how that happens in a bacterial cell, and as you can see, it's really straightforward. You have DNA floating around in the cell, transcription occurs making mRNA, and the mRNA can move directly to a ribosome, and translation occurs making a polypeptide. As usual, processes in eukaryotic cells are a little bit more complicated. Remember that the DNA is located inside the nuclear envelope, so transcription takes place in the nucleus. Using DNA as a template, a strand of pre-mRNA is formed, and then in eukaryotes there's a step called RNA processing. This can mean that the mRNA is cut, that little snippets are added to the ends, and now you have a strand of mRNA that's ready to leave the nuclear envelope and go out into the cell where the ribosomes are located, and then translation can occur, creating a polypeptide. So here you can see we have the same central dogma. DNA goes to mRNA via the process of transcription, and mRNA goes to a polypeptide via the process of translation, but of course it's just a little bit more complicated. So here's a thought question for you to think about. We know that DNA is made up of 4 nucleotides. That is, there are 4 letters that DNA can write in, A, T, G, and C. We also know that DNA codes for amino acids, and so we can use a certain number of letters to code for specific amino acids. So, how many different amino acids could DNA code for if each nucleotide coded for one amino acid? Well, obviously the answer to part A is 4. If you only have 4 letters and takes 1 letter to code for an amino acid, you could only code for 4 amino acids. Now, what if we used pairs of nucleotides? Here, the answer is 4 times 4, or 16. So, the DNA is arranged in a triplet code, a series of non-overlapping 3 nucleotide words. So, for example, any stretch of DNA that has A, G, T is going to code for the amino acid serine. Now, remember that DNA is a double helix. It's made up of 2 strands. So here you can see at the top an example of the double helix. And if we zoom in on part of the double helix, we see 2 strands of DNA. One of them is a template strand. So the template strand in this example goes A, C, C, A, and so on. Therefore, when transcription takes place, we're going to make an mRNA strand that goes U, G, G, U, and so on. Remember that mRNA, or all RNA actually, has U instead of T. So U is the nucleotide that pairs up with A in RNA, just like T is the nucleotide that pairs up with A in DNA. And then, once you have those series of nucleotides in the mRNA strand, each set of 3 nucleotides forms a codon and will code for one particular amino acid. And so at the bottom, you can see the polypeptide or protein that will be formed using that particular sequence of mRNA from that particular sequence of DNA. As I mentioned, in RNA, T is replaced by U, so A pairs with U. And we also talked about there are 64 possible triplets, 64 different words that you can make with a combination of 3 letters. Now, 61 of these code for amino acids, and 3 of them don't code for any amino acid. Those 3 are called the stop codons, and they signal an end to translation. It's very important to remember that no codon can specify more than one amino acid. So a particular set of 3 letters can only mean one particular amino acid. That's really important in making sure we get the right proteins. If you had a codon that could specify, say, either tryptophan or serine, then your proteins would be all over the place. So it's really important to remember this. Each codon can only specify one particular. This is just a diagram showing all 64 possible codons and what they code for. You definitely don't need to memorize this whole diagram for this class, but you do need to recognize that there are some amino acids that are only coded for by one particular codon. For example, tryptophan is only coded for by UGG. And there are some amino acids that can be coded for by up to 4 different codons. So CCU, CCC, CCA, and CCG all code for the same amino acid. You should also notice that there are 3 codons, UAA, UGA, and UAG, that all code for stop. There's no amino acid associated with those codons. They just mean we're at the end. And one last thing to check out here, AUG can either, it codes for methionine, but it's also used as the start codon, the point that shows where translation should begin. All right, so take a look at the following section of DNA. Now, this is the template strand, and it reads ATAACGGCTCATGTA. So the first thing you should do The genetic code is basically universal, except for a couple of rare exceptions in some obscure archaea. All organisms on Earth use the exact same code, that set of 3-letter words to code for a particular set of amino acids. What that means is that a gene can actually be taken out of one organism, put into another organism, and then it will be transcribed and translated correctly. Here are some examples of that. This is a tobacco plant expressing a firefly gene, and a pig expressing a jellyfish gene. This is exactly the same thing you're doing in lab when you carry out bacterial transformation. We're using a gene from a jellyfish, just like is shown here in the figure, and popping that So let's look at transcription, how RNA is made using DNA as a template. So here we see all of the steps in transcription. Up at the top, we see the location of the promoter. That's the point along the DNA where the RNA polymerase grabs hold. And the RNA polymerase is represented as that sort of guitar pick shaped translucent molecule there. The RNA polymerase in initiation grabs onto the DNA, unwinds just a tiny section of it, and starts building RNA using the template strand. The next phase is elongation, where the RNA polymerase just basically scoots along the DNA, creating a strand of mRNA as it goes by popping in the RNA nucleotides that pair with the template strand of DNA. And the final step is termination, which is basically just where the RNA polymerase falls off the DNA strand, and then transcription is complete. If we zoom in on what's actually happening there with the RNA polymerase molecule, you can see how the RNA polymerase molecule is pulling in RNA nucleotides, matching them up with the template strand of DNA, attaching them to the growing RNA strand, and then continuing on downstream, moving along the DNA double helix.